is going to be attending Case University this fall, and he was once a student at High School for Medical Sciences and Science in the Bronx, and until he moved to Pleasantville High School. So welcome, Reginald.
As she said, my name is Reginald Anyichie, and over the past 2 summers, I worked with HCS. I worked with Dr. Oliver Hubbard, and I worked at the Columbia University. And the title of my presentation is Characterizing Mutations Affecting Neuron Formation of C. elegans. So let's start. Here it goes. Basically, we're going to start with what mutation is. Mutation is a permanent change in the DNA sequence of a gene. So basically, like 2 years ago, when I first came to HCS, I found out C. elegans worms went through a process called mutagenesis. This process basically involved isolating mutants affecting the development of neurons. So basically, last year in 2007. This research was focused on characterizing the mutations that were isolated. Basically, the general purpose of this this research is to determine how different neurons are specified during development. Am I going too fast? Yes. I'll go back. Sex organs. Basically, in these worms, hermaphrodites produce both octites, which are also known as ovaries, and they produce sperm. They can fertilize themselves, like they can impregnate themselves without having sex with the opposite sex. And they cannot mate with each other. Basically, 2 hermaphrodites cannot mate with each other. They either have to mate with a male or they produce by themselves. Yeah, this is basically the structure of hermaphrodite. This is like the female hermaphrodite. Just the hermaphrodite, my bad. Males, well, there's not that much info about males because they arrive at a lower frequency than hermaphrodites. And basically they can fertilize hermaphrodites too, yeah. And this is the organization of a male, as you can see. Yeah. Basically, we screen for, these little dots you see here on the picture, those are the different neurons. One of them are ASH, ASI, ALY, and PVQ. Basically, like this diagram here is like what we did over the summer. We took like one worm, like one hermaphrodite, which is the parent. We put it through the process of EMS, which is mutagenesis. Then after a couple of days, we stored them inside this plastic container, and they like cultivated. So we took the next generation and separated them into different plates, and that's what we continue to do over the summer. Basically, we screened the second generation of progeny for loss of ALY neurons. The specific aim of this project. Throughout this project, 19 mutants were isolated during screening. So basically, we want to determine which genes they are affecting. The materials that were used in this project were basically agar paste, which is like a small container plate where we store the bacteria, pig sticks, and GFP microscopes. The methodology is pretty short and simple. Yeah, we first tried to determine which chromosome one of these mutants were affecting. And for that, we just crossed the hermaphrodite with the wild type males, and yeah. We analyzed the cross progeny, and this led to the conclusion that hermaphrodites were wild type, and the males were mutants. In conclusion, we found out that this mutant is located on the sex chromosome X. Also, one candidate of the X chromosome, one candidate on the X chromosome is a transcription factor. So for future studies, we will sequence it to see whether it is mutated. And if it's not mutated, if the transcription factor is not mutated, we will determine more precisely where the mutation is located on the X chromosome to find the gene affected. These are my references, acknowledgements. I'd like to thank God, Dr. Sat, my mentor, Dr. Oliver Hobart, my co-mentor, Vincent, the lab crew, MSKCC, Columbia University, ACS staff members, everyone else that's here. Thank you. Any questions? Yeah. Any more questions?
I have a question for New Hampshire.
Yeah.
I have a question from New Hampshire. Gene is on the sex chromosome X.
Basically, we process it through EMS. I'm sorry. Basically, process it through EMS.
Okay.
Yeah.
How do you make the mutants?
Huh?
Like here? Okay. So my question is, how do you know that the gene that you're looking for is on the X chromosome, the sex chromosome?
We looked at it through the microscope, the GFP microscope to see if there's any missing neurons. Yeah, so that's basically it. Any more questions? She had a question. I'm sorry. Oh yeah, you? I just did.
We're not going to do that. I'm just briefly in 2 or 3 sentences, but not slide by slide.
All right. You could copy. Oh, yeah. Yeah.
In the plates, you were only using the hermaphrodites, or inside the plates were there the hermaphrodites and the males, or was it?
Yeah, sometimes we'll leave only the hermaphrodites. Sometimes we'll mix if we want to get more males, we'll put the hermaphrodites with the males. If we wanted to get only hermaphrodites, we just leave the hermaphrodites to reproduce on its own. Any more questions?
We'll take 2 more questions and then we'll have to.
Yeah. I didn't see it, sorry.
Okay, so when hermaphrodites is made,
So when the hermaphrodites mate, they only produce hermaphrodites, or when the hermaphrodites and the males mate?
No, hermaphrodites don't. Wait, could you say that again? I'm sorry.
When 2 hermaphrodites mate, they only produce hermaphrodites.
2 hermaphrodites cannot mate. Only a hermaphrodite and a male can mate.
So why would you put them in the petri dish or whatever?
Because hermaphrodites, as I said earlier, they both have male organs and sex organs. Inside, it's like when they combine.
I remember what you said. But I still have another question. When the males and the hermaphrodites mate, only males are produced?
Yeah. Any more questions? You at the back.
what's your future?
Future studies? Sure.
Thank you.
Yeah. Any more questions? I don't have any more questions. Thank you.